Good afternoon, everyone. My name is Tong Yen Wah. I'm from the National University of Singapore, and I'd like to welcome all of you to the webinar at NUS that is under the Singapore International Water Week. We are all from the NUS Environment Research Institute. I'm also from the Department of Chemical and Biomolecular Engineering. I would like to thank you all for being able to take part in today's event. We hope it will be interesting webinar for you and hope that you'll be able to participate with questions and comments. We would like to ask if you have any questions after the presentation. You can type it into the Q&A chat rather than in the chat box. So use the Q&A function. You can vote for similar questions that you'd like to see, and those that get upvoted, we'll go through them first. So please feel free to key in your questions at any time. Right, so let me introduce today's speaker for the webinar. It is Dr. Sheetal Parakh. She is a research fellow now in the NUS Environment Research Institute, and she completed her PhD, obtained a degree last year. So she has been working in the areas of microalgae, that's the topic for today, by developing photobioreactor technologies. She has studied the culture and optimization of microalgae and also characterization of the microalgae for various applications. So hopefully today's application will be of interest to all of you. I won't take up too much time, so let me invite Dr. Parakh to give the webinar today.
Thank you, Prof. Tong, for the introduction. I will share my screen now. Hi, everyone. Good afternoon. Thanks for joining us today. Today's topic of my presentation will be biotransformation of food processing wastewater into microalgae-based high-value products. Now, the question is why we are specifically talking about food processing wastewater. It's because of the rising food demand. The global demand for food is actually projected to increase by 25 to 70% by 2050. And this is because of the population growth, economic growth, also the rising calorie consumption. To keep up with this rising food demand, our food production industries and food processing industries will also have to step up their output. And while doing this, they might also increase the water consumption. Now, food processing industries consume a lot of water compared to the other materials. Also use water to clean their raw materials, also to wash the equipment. So if we take a look at these certain examples that I have put it on the screen for you, if you look at the soybean processing industries, it actually generates around 7 to 10 liters of wastewater. Now, soybean is a main ingredient in soy products like tofu and soy milk, which is quite popular in Asia. And you can imagine how much wastewater can be generated if with the rising food demand, with the rising of demand for the soy products as well. The wastewater in the dairy industry actually can range around 0.2 to 10 liters per kg or per liter of the products that they're processing. In the starch processing industry, it can go up to 20 liters, and same for meat processing industry, which can consume up to like 60 liters, can produce 60 liters of wastewater. So what this wastewater constitutes? The wastewater actually from food processing industries is quite unique. It contains a lot of organic components, which is represented here in the form of chemical oxygen demand, COD. So you can look at the numbers. They are all in like few thousands. Then the food processing industry wastewater also contains a lot of nitrogen and phosphorus. Since it's a food, all these good nutritional components which are actually getting wasted in the wastewater could be problematic when we want to discharge this wastewater. So we definitely cannot leave it without any treatment. Now, what are the current treatment methods? The first treatment method that is very popular is aerobic treatment. Now, what happens in the aerobic treatment is that the wastewater is subjected to aeration, and there are also aerobic microorganisms inside the system which break down the organic pollutants. So although this treatment quite efficiently break down the organic molecules, but they cannot take so much of nitrogen and phosphorus, so the effluent that comes out from this treatment contains a significant amount of nitrogen and phosphorus. While microorganisms are breaking down the organic pollutants, they also release carbon dioxide. That's another negative point for this treatment method. And they create sludge. So sludge is the bulk of microorganisms that are growing in the system while treating the wastewater. And to keep the operation stable, we have to frequently discharge the sludge from such systems. So this is about the aerobic treatment. The second treatment method for treating wastewater is anaerobic treatment, which is getting popular. The reason why anaerobic treatment it's getting popular over aerobic treatment because of low operational costs. You see, in the aerobic treatment, you have to provide air for the microorganisms to break down the organic pollutants. While in the anaerobic treatment method, as the name suggests, there is no oxygen. That's why you don't have to provide any extra air, and it cut down on the operational cost. Also, the another interesting thing about the anaerobic treatment is that you can produce biogas. So there is some resource recovery from the treatment method. And but both of these methods, to certain extent, we cannot fully recover high value products from these two methods. And that's where our solution comes in. What we are trying to do here is to cultivate microalgae in this wastewater. So I'll come back to this slide, but first I will explain more about microalgae. So microalgae are like photosynthetic microorganisms. So they are just like plants. In the presence of sunlight, they can fix carbon dioxide into a biomass, and they grow in wide range of habitats. By wide range of habitats, what I'm trying to say is that they can grow in fresh water, they can grow in marine water, they can grow in cold regions like Arctic, or they can even grow in tropical regions or deserts. There are more than 50,000 microalgae species which are present on the planet Earth. And all these species are ecologically different, metabolically different. So there is quite a lot of applications that can come out of microalgae production. Now, I said that microalgae are quite similar to the plants, but they are kind of superior. Why? Microalgae actually do not require any arable land, so we can cultivate them anywhere. They have short life cycles, which can only last up to a few days. So, which means that we can actually do a lot of production within a year. They have higher photosynthetic efficiency. And the last but not least, they have very high nutritive value because their biomass is packed with lipids, proteins, carbohydrates, vitamins, and minerals. Now, since the biomass has so many nutritional compounds, microalgae actually finds application in many industries, starting from functional food. Now, what is functional food? It's where you actually take a normal food product and include additional nutritional substances. So instead of doing that, we can actually incorporate microalgae biomass into such products for example, ice cream, pasta, noodles, and increase its nutritional value. We can also extract protein from microalgae and convert it into alternative meat or alternative dairy. Companies like Impossible, Beyond Meat, they're actually extracting plant-based proteins, for example, pea protein or soy protein, and converting them into the alternative protein. Now, microalgae protein could also be a good option for such industries. Microalgae also contains like pigments and omega fatty acids, which are very good for personal care and nutraceutical sectors. Recently, microalgae has also been popular as biostimulants. So what biostimulants are, they actually help plants to take nutrients effectively, and they also increase the tolerance of plants towards abiotic stress. So that's an emerging area. Microalgae are also popular as fish feeds or dog foods. So currently, like there are more than 100 biotechnology companies worldwide who are working on this amazing microorganism, and they are producing different products. Now, the question is that if the microalgae is so interesting and it has so many applications, why it has not been widely implemented? There are many challenges in the process, but for today's talk, I'll be restricting myself to one challenge. That challenge is raw materials, securing raw materials for microalgae. So let's look at the microalgae recipe. To produce 1 kg of microalgae biomass, we require 4 to 2000 kg of water. Now, this is a wide range. The reason behind the wide range here is that microalgae can be cultivated in 2 different cultivation systems. The first is autotrophic, where they photosynthesize. But here, the photobioreactors that are currently available can maximum produce 10 to 15 grams per liter of biomass, which is why you can see the numbers going, the water quantity going higher. There are also other kinds of system like heterotrophic cultivation. Some microalgae can be actually cultivated heterotropically, where the biomass concentration can go up to 100 to 200 grams per liter. That's where you can actually see the water quantity going towards the lower side. But anyway, in both cultivation system, water actually plays a big role. The other ingredient in the list, the next ingredient in the list would be carbon. So carbon actually is like 50% of the dry biomass. So that's why we need a lot of carbon to grow microalgae biomass. Now, if you're growing it autotropically, one can claim that we can use carbon dioxide from the atmosphere, but the concentration of the carbon dioxide in the atmosphere is only 0.04%, which is pretty low because most of the microalgae need 1 to 5% of carbon dioxide for proper growth. So we have to concentrate the carbon dioxide from the atmosphere, which could be cost intensive. We can use industrial flue gas, but there are certain other problems which I will not go into detail for now. The next kind of a carbon source, if you are cultivating in terms of like if you're doing a heterotrophic cultivation, you can use organic carbon like glucose. But if you start using glucose to cultivate microalgae, it will become very expensive, and that's actually a very big problem. The next ingredients in the list are actually nitrogen and phosphorus. Now, both of these ingredients are actually part of fertilizer. So if we start using these ingredients, synthetic ingredients to cultivate microalgae, we would be directly competing with the agricultural fertilizer demand. So this gives you a picture that how securing raw materials to grow microalgae is actually an issue. And that's why this is one of the many reasons why microalgae is not widely cultivated so far. So coming back to our solution, what we are looking at here is that there are 2 problems. We have one problem where we have food processing wastewater, which contains significant amounts of organic carbon, nitrogen, and phosphorus, and it needs to be treated before discharge. On the other hand, we have microalgae cultivation, and the problem there is securing the raw materials. So why not we integrate these 2? Now, there are different kinds of wastewater that we can use to cultivate microalgae. Why specifically food processing wastewater? The reason we chose this is because food processing wastewater is quite safe. It doesn't have any toxic material, it doesn't have much heavy metals. So if the biomass that we are cultivating from this wastewater could be used for high value applications. So this is our solution. Now, before I get into the projects that we have been doing, I would like to give an overview of the work that we do. So what exactly we do? We actually like scout for waste materials, so local food and beverage companies' waste materials. We characterize them. If required, we pre-treat these materials and make them suitable to grow microalgae. After that, we use these waste materials in our cultivation systems, and we optimize the conditions. We harvest this biomass, we process it, and then we are converting this into the products. So today I'll be talking about two such case studies that we have been working in the past few months. So the first, the case study that we've been working is actually soybean processing wastewater. Now, soybean processing this wastewater actually we collected from a local soy milk manufacturer. So I would like to quickly run you through the process to understand where we got this wastewater from. So in the soy milk industry, the soybeans are actually washed and soaked. So that's a source of wastewater. Then they are actually grinded so that they can remove the soy milk from there. And after grinding, the equipment is actually washed. So that's where the second source of wastewater comes from. Then it goes to a cooking or a sterilization stage. And after this, again, the equipment will be washed. And then that's where the wastewater comes from. So at every step, there are different sources of wastewater that are coming. For this study, we actually collected wastewater from the grinding stage and also from the sterilization stage. So what we did was we actually brought this wastewater back to our lab, and this is how it looks. So it's like milky since it's coming from a soy milk factory. We allowed it to sediment overnight, and after sedimentation, we actually got a very much clarified soy wastewater. But it contains like very small suspended particles. So we subjected to filtration. We used 0.45 micrometer and 0.22 micrometer filter. And then our end product was a sterile and suspension-free soy wastewater. We characterized this wastewater and found out that the pH for both of these wastewater was around like 5 to 6, and the organic content, nitrogen and phosphorus of this wastewater was quite high, specifically for the grinding stage. So this kind of proves that we definitely have to treat the food processing wastewater before we discharge, looking at these concentrations. So what we found was we started cultivating the microalgae in the soy wastewater directly. The only one thing that we did was we changed the pH from 5 to 5.7, which was the original pH, to 7, because microalgae can grow better between 6 to 8. What we found was the soy wastewater, which was collected from the grinding stage, actually gave a highest biomass concentration compared to the control, which started going to a stationary phase around 0.9 gram per liter, whereas the microalgae cultivated in the grinding stage went up to 1.7 gram per liter. So it was quite high. And again, I would like to emphasize on this point that this was just waste. If we do not use it for this purpose, it will just be disposed without being able to recover these nutrients. So the next thing that we looked at was the protein content in the microalgae biomass that we were cultivating. So the protein content was actually quite comparable to the control experiment for the first 4 days, and later the control experiment, the protein content in the control experiment started dropping, whereas the soy wastewater protein content was still like higher than the control experiment. So this actually shows again that we can create the similar amount of protein without using any synthetic chemicals. The protein yield was actually quite high in the soy wastewater, and this is because that the biomass in the soy wastewater was significantly higher than the control. So you can actually see the higher protein yield here. Now, looking at the nutrient removal efficiency, what we found that at the end of the experiment, around 20 to 50% of carbon, nitrogen, and phosphorus were removed from the soy wastewater. So this shows the potential of microalgae in removing or in treating the soy food processing wastewater. Now, what we are trying to do is that we would like to further boost the nutrient removal and also the biomass production in soil wastewater. So we have decided to do some pre-treatments. We hypothesize that the soil wastewater actually contains some complex molecules which are difficult for microalgae to digest. So we would like to break down these molecules into simple compounds, which microalgae can then intake. And this way, we could increase its biomass production and also improve the nutrient removal. Our future plan is actually to demonstrate this technology at a 100-liter scale. We have designed a 100-liter photobioreactor, and we are currently constructing this. Uh, we've received a funding from Technology Acceleration Program NUS to build this reactor. Now, let me take you to the second case study that we have been looking at. It's from food waste digested. Now, this is not exactly a food processing wastewater, but also a very interesting kind of a medium that can be used to grow microalgae. Now, food waste is a very important problem in Singapore. Singapore generates around 740,000 tons of food waste per year, and the recycling rate is around 20%, so it's quite low. So what my colleagues at E2S2 Create are doing is that they treat this food waste using anaerobic digester, and they are generating biogas and digest it out of it. Now, this digested, if you separate into solid and liquid, the solid part can be used to grow vegetables as like a biofertilizer, but still the liquid digestate needs to be treated. This is where we came in. We collected this digestate from our colleagues at E2S2, and this is how the raw digestate looks like. This is before the solid liquid separation. Since it's actually very dark and a black liquid, so we centrifuged it. We did try to do sedimentation, but it was not possible to separate this through the sedimentation. That's why we use centrifugation. After centrifugation, we could remove most of the solids, but there were still more suspended particles that remained in the digestate. So you can actually look at the creamy color here. So we did step filtration, so use different sizes of filter, and finally we were managed to get something that looks like this. So this we characterize this digestate, and its pH was around 9. It also contained a high amount of organic carbon and ammonium and phosphate. Now, the reason that I am actually mentioning ammonium and phosphate here instead of total nitrogen and total phosphorus is because this digestate comes from the anaerobic digestion process. During the anaerobic digestion process, most of these complex molecules are already broken down. So that's why we can only see the simple molecules here, which are very easy for microalgae to consume. Now, there was one problem here though. The digestate, although we filtered it, it looks something like this. So it's microalgae are photosynthetic microorganisms, so they need light. If the digestate has this kind of a color, there is a possibility that when we grow microalgae in it, there could be light limitation, and which will hamper their productivity. Hence, what we decided to do was we diluted the digestate. So this is a 100% digestate, and then these are different dilutions. So this is 50%, 20%, 10%, 5%, and so on. So you can actually clearly see that as you dilute, you can the transparency of the digestate increases. Now, this was also proven through the UV vis spectrum, where we measured the absorbance across a wide range of wavelength. And we found out that the 100% digestate was actually blocking the photosynthetically active radiation region, which is 400 to 700. So it actually blocks this region quite a bit, between 400 to 500. The 50% was also was blocking the region. So when you go down like 20%, 10%, the blockage due to the color actually starts becoming less. So we decided to cultivate our microalgae in 20% to 1% diluted digestate to see if it can grow. Now, to our surprise, the microalgae actually did not grow in any of the diluted digestate. So if you can see here, the control experiment actually went up to like 1 gram per liter within the 7 days. whereas the digestate microalgae grown in the digestate did not show much growth. It's also clear from these picture here. These are the control experiments, and these are the microalgae grown in digestate. So no growth was observed. So we took a step back and we decided to look into the composition of digestate, and we compared this with the growth media. And what we found was very interesting. The digestate actually contains four main components, which is sodium, potassium, ammonium, and chloride, while the other components which are present in the growth media were actually lower than the medium itself. So when we dilute the digestate further, this gets even further diluted, and this could explain why our microalgae could not grow in the 20% to 1% digestate. So what we did here was we started supplementing the digestate with nutrient. So what we did is that we broke down the growth media into three components. The first component is represented by P, which actually means phosphate. The second component is represented by S, which actually means the salts of magnesium and calcium. And the third component is trace metals, which actually represents the other metals like copper, cobalt, iron, which are actually present in the growth media. So we used a 10% diluted digestate and then supplemented the digestate with these components in different combinations. And we found out that when we supplement the digestate with all three components, P, S, and Tm, we could see the very good microalgae growth, and within 10 days, it reached 1.5 gram per liter, while the control was still around 1 gram per liter. And with other combinations, actually, the microalgae growth was not very significant, except the this part, which is salts plus the trace metals. The another interesting thing about this experiment was when we supplemented the digestate with all these components, all of the ammonium that was present in the digestate was removed by day 10, was by the end of this experiment, which is quite interesting. So although we try to focus on this aspect, that we try to get most of the nutrients from the waste resources, sometimes it's not possible because different waste have different components, and we have to adopt to them. We have to develop certain strategies to work with them, and that's what we have been doing here. So after this, actually, what we would like to do is we would like to try growing the microalgae in a higher digested concentration, because when we are growing in 10% digested, we are actually diluting it with the fresh water, and we would like to avoid doing that. So so okay, wait, let me first get into the protein content part. We compared the protein content with the control, and we found out that at every stage, the digestate microalgae grown in the digestate had very high protein. On the day 6, it even reached 68%, which is quite amazing. And it's significantly higher than the control. So again, although the most of the components between these two experiments were same, except the part that the nitrogen source in the control actually came from the nitrate, whereas the nitrogen source in the digested experiment came from the ammonium. Everything else was same, and still we actually got a very good result with the protein content. And the protein yield was also very high in the 10% digested experiment. Now, as I said before, that we would like to work with higher percentage of digestate. So this is where our ongoing work is looking at. What we did in a preliminary experiment that we treated the digestate with activated charcoal, different concentrations of activated charcoal, and we found out that by doing so, we can actually remove these colored compounds from the digestate. So you can actually see in this picture that this is a control experiment where no activated charcoal was added, and these two were treated with the activated charcoal, and then the color was actually gone. We also looked at the UV spectrum here, and you can see that the the blockage in the 400 to 700 region was only high in the control, while with activated charcoal, we could bring it down significantly. So we will be actually going after this idea. We would also be trying different kinds of biochar, since activated charcoal could be expensive for a pre-treatment. So we'll try with different biochars and see if we can remove the color from the digestate. And in that way, we will be able to cultivate our microalgae in undiluted digestate. So this brings me to the summary of my presentation. And I would like to again put emphasis on this solution. So we actually have waste resources locally available. We could just use them to grow microalgae, and in return, we can get biomass, which has multiple applications, and we could treat the water. So this is our one step towards circular economy. So at the before I end my talk, I would like to thank our grantors. I would like to thank Technology Acceleration Program NUS, which is supporting this study. We would also like to thank our industrial partner, Mr. Bing, for providing us with the soybean processing wastewater, and our academic partner, E2S2, for always helping us with the digestate. Thank you so much. If you want to connect with us, or if you want to know progress on our project here, please feel free to reach out to me or Professor Tongenwa. Our email IDs are displayed on the screen. Thank you so much for joining today.
Great. Thank you very much, Sheetal, for the very clear presentation. Gave a very good and detailed description of the work. So I'd like to now open the session for discussion. I'd like to invite attendees to ask any questions. If you do have, please do use the Q&A function in the Zoom webinar, and we will ask the questions as they come up. All right, we do have one question first. The question is, to avoid the dilution or activated carbon treatment of liquid digestate to get light, why not use microalgae species that is chemoautotrophic other than photoautotrophic?
Yeah, I think I can answer that. Yeah, so thanks for the question, Hylene. Actually, we can definitely do that. We can use microalgae species which are heterotrophic in nature, and then the color of the digestate wouldn't be an issue as long as they can tolerate the high ammonium concentration. And I'm definitely going to try this. Actually, this is gonna be my next part of my study after I finish working with the activated charcoal. And the reason that we also have to work on the activated charcoal treatment is because not all microalgae can be grown heterotropically. So the activated charcoal solution could be more universal than the growing them heterotropically.
Great. There's another question in Q&A that is, it says, is this an interesting study? And the person is keen to know if the soybean wastewater was used directly, or do you need to add any nutrients?
The results that I presented here, we did not add any nutrients. We only changed the pH, which was from 5.7 to 7. So that's all. After that, I just filtered and used it directly.
Good, good. Okay, we have a third question. This is on the biomass. So the question is, what will be the outcomes of the biomass that's produced, and is it safe to discharge directly?
The biomass here, we are not planning to discharge. We would like to use it. The current application that we are thinking about is protein. So we are not sure about if we can use it directly in the food or feed applications. For that, we would have to do certain toxicity studies, also some microbiology tests to see if there are any other contaminants. So, so far, since we are doing this under sterile conditions, for both the cases, I have actually filtered them with 0.22 micron filters. So the pathogens are removed. And mostly the soy processing wastewater should not contain any other toxic contaminants, which could be a concern for us. But I think I would be able to tell more confidently once I have the test results. But yeah, our applications would be into food and feed.
Great. There's another question. Are these processes temperature sensitive? So meaning that other than in the tropics, how effective will these work?
Thank you. So actually, microalgae are very ecologically diverse. So there are a lot of species which can grow in different regions. So if you are working in a region that is not tropical, you could actually look at the native microalgae species. So a lot of researchers, they try to isolate the local species and grow them because they're already growing in the nature. So which means that it would be easy for us to apply them in our studies. And other than that, if you try to grow them out in the outdoor conditions, but if you want to grow them in indoor conditions, this shouldn't be an issue because we can control all the cultivation conditions, including the temperature.
Right. Good. So there's another question. So since the produced microalgae concentration is only several grams per liter, how will it be extracted from the culture medium? And can the culture medium then be directly discharged or will it be recycled? And then the add-on is the used medium safe to be discharged?
Yeah, okay. So definitely the concentration here is only going to be a few grams per liter. So the bioreactor that we are trying to build will actually have in situ harvesting system attached to it. So we would definitely concentrate it. And then after that, we will reduce the volume. And then for now, we are thinking about centrifugation as a treatment method because the products that we are trying to get out of it are high value. So if we use energy intensive operation at this point of time could still be okay. But in the future, we will definitely have to see how we can increase or decrease the energy intensive processes that are used for harvesting. Yeah, but for now, I think we would stick to centrifugation.
Is the medium then safe to be discharged?
The medium, okay, so for the soybean study that I did, currently the level of organic carbon, nitrogen, and phosphorus is still higher than the discharge guidelines. So we cannot discharge it. But in my next part of my study, what I'll be trying to do is that I'm going to use certain pre-treatment methods to break down the complex molecules which were not consumed by microalgae when I used it now. So in that way, we would try to reach the discharge limits and then it will be okay to discharge. For the digestate experiment, I guess the ammonium was mostly removed. So I think we can discharge it after that.
Thank you. Are there other questions? If you do have any, don't be shy. Please just type it in the Q&A box. While we wait, I do have one question. You did not actually talk about the species of microalgae that you have used here. Are they freshwater species or are they marine microalgae?
Yeah, the one I'm using is actually a freshwater species. It's from Chlorella family.
So for that, when you're using the food waste digestate, the salt concentration is quite high, right? Would it be suitable or better to use marine species rather than freshwater species?
Actually, the salt concentration in the digestate was around 3,000, like the chloride was 3,000 ppm. And the microalgae species that I'm using is quite comfortable with it. I have even tried to grow them in higher concentration. It was okay. Because the marine species, I don't know if they can grow in the 3,000 ppm concentration because they are used to 20,000 ppm of chloride. Yeah, so I think the freshwater is still okay for the digestate.
Okay. There's a separate question that is in the chat box. Is do you foresee any issues for scaling up of this process?
For the scale-up part, so for the soybean wastewater, because it contains organic carbon, it could be easy to scale up. The only thing would be contamination, because we are going to grow this mixotropically, where we will use light and also the organic carbon. So contamination could be an issue. And for the digestate experiments, I think the one issue would be they have organic carbon inside, which cannot be used by microalgae. They are not biodegradable, at least not degradable by microalgae. So would still need further treatment. So these two issues that I can see at the current time.
Great, thank you. Would the attendees have any other questions about the work done here for today? Okay, one question came up. It says, thanks for the interesting talk. Have you planned to evaluate the cost effectiveness of doing the different wastewater purification process? And are you also planning to do a pilot and industrial scale?
Yes, we are actually planning to do this at a pilot scale. The cost effectiveness study, I think, would be done when we do it at the pilot scale, because with the lab scale data, it's kind of difficult to project the figures. Yeah, but we are building a 100-liter photobioreactor, and it should be ready in a few months. So once that we get it and it's running, I think we will definitely look into this. Thank you so much.
We still have some time if you do have any questions. If there are no other questions, I guess it's a Friday afternoon after all. Oh, there's one. Oh, it's not too late for Friday afternoon. We can still have one more question. Can we only use the biomass generated for protein production?
No, we can actually use it for, it just depends on what application you're looking at, because that will decide the microalgae that you are selecting. So the microalgae that I selected is actually protein rich. But if you want to grow if you want to grow them for biofuels, you can actually choose a microalgae which is lipid rich. So there are different, depending on your application, we could actually design the whole thing again.
I guess we don't want to keep the attendees also, but if you do have questions, I guess you can always contact the Sheetal directly too, through the email. But another question came up, so we can answer this. Thank you for the talk. What are some of the limitations with this process? For example, particular chemicals present in the waste that would cause the process to fail?
The limitation wise, I guess it all depends the tolerance level of microalgae. So if there are certain compounds that are present in the wastewater that or their concentration is very high for the microalgae, then this might fail. So I think for that part, it's more of we need to try first, since waste are very complex and we don't know what is inside. So it's always will depend on the trial.
Right. So again, Sheetal did post her email address earlier. So if you manage to catch that, you can always ask her, contact her for more information. But if not, I would like to thank everyone for joining the webinar at NUS for today, and hope you will take part in the continuing seminars next week also. So I'd like to thank Sheetal for giving the very interesting and clear presentation, and hope that everyone has managed to find some useful applications for microalgae after listening to this. Thank you all, and hope you all have a great weekend coming up. Thank
Thank you
you.
so much.